Okay, so I'm going over the 7.2 homework. This is the first question that starts with cell organization, the relationship between cytoplasm and the nucleus. The nucleus is inside of the cytoplasm. It's not part of the cytoplasm. The cytoplasm is the jelly-like substance that kind of suspends all of the organelles, but both of them work together to keep the cell alive. Organelle literally means little organ. So if you think of all of our little organs that make up us as a cell, then all of the little things that have different functions will make up the entire cell. In the visual analogy, the nucleus is going to be the office. It's going to hold the blueprints. It's illustrated by those people at the desk. The nucleus starts the message, and then it takes it to the RNA, which actually can leave the nucleus and go to another place in the cell. The feature of the nucleus does not clearly show the chromatin. We'll get into that in second semester. Another possible analogy that can be compared with structure and function of an airport control tower, because they're telling something. So, you could do that however you wanted to. Vacuoles. Vacuoles are going to be sac-like structures that store materials, very similar to vesicles. Then two roles of the central vacuole in plant cell is going to store materials. It's also going to support the cell. The central vacuole is very much going to be full of water. It's going to help that keep turgor pressure, which we're going to talk about a little bit later too. Contractile vacuoles are different from other ones because they're actually pumps. They're going to pump the water outside of the cell. Usually all the vacuoles are going to hold storage things. In the diagrams, it asks you to label the things. The top one is centrioles. Centrioles are going to be utilized only in replication of the cell. The next one is the lysosome. The lysosome is going to be the custodial workers. They're going to clean up and break down things. They have enzymes inside that help to break things down. And then the central vacuole is in the plant cell. You knew that it was a animal cell on the left because it has a circular outer rim, and it's a plant cell on the right because it's very boxy and it's got that structure due to the cell wall. The role of lysosomes, we already talked about. They're going to break down the molecules in waste. They have enzymes inside that break down things. Without them, obviously waste would build up in the cell and it would cause cell death. Structures of the cytoskeleton are in animal cells but not plant cells. Those are the centrioles. Again, I said it's going to be used in replication. We are going to use those to maneuver those microtubules around the cell. Other structures in the cytoskeleton are similar to cilia. Cilia are finger-like projections that help to move things along. Number 13, ribosomes. Ribosomes are going to be the protein factories. The RNA is going to travel to the ribosome so that they can assemble whatever protein that is needed to be made. Lipid components of the cell membrane, that gets made in the endoplasmic reticulum. That's going to be like a transport system. The difference between rough ER and smooth ER is rough ER has the ribosomes studded on them, and smooth ER has no ribosomes. Using cell factory as an analogy, the Golgi apparatus is going to be like the custom shop, or like UPS. It is going to put the finishing touches on proteins before they leave the factory. A cell's Golgi apparatus doesn't function properly. How can this affect the cells? That means it's not ready to be received by other things. They don't have their correct proteins in there. The proteins that are going to the spot that they should be will not help them function properly. Then you had a Venn diagram. Chloroplasts and mitochondria are in cells. Chloroplasts are only in plant cells, but mitochondria are in both. Both of these were part of the endosymbiotic theory that you read that article on. Both of them have a double membrane. Both of them have their own genetic information, and we see that both of them descended from other microorganisms. If we focus on the left on just the chloroplasts, we said they were only in plant cells. They are going to capture the sun's energy, and they have chlorophyll to help them go through the process of photosynthesis. Mitochondria are in both of those cells. They release energy and cellular respiration. For 19 through 22, it was true and false. Chloroplasts are never in animal cells. That is true. Unlike chloroplasts, mitochondria are surrounded by a double membrane. That is false because they are exactly the same in that aspect. Nearly all of the mitochondria in your cells were inherited from your mother. That is true. Number 22 is false. Both chloroplasts and mitochondria contain their own genetic information. When we look at the cellular boundary, this is something that we're going to get into a little bit later as well too. Most cell walls are porous to water and other materials, but strong enough for support. Nearly all of plant tissue called wood is made up of cell walls. Besides supporting and protecting a cell, the cell membrane regulates what comes in and enters. It's the security system. The diagram, that first top black chain is the carbohydrate chain. It is on the cell lipid bilayer. There's 2 layers of lipids. That's why it's called a lipid bilayer. The top is the hydrophilic heads. The bottom is on the hydrophobic tails. And then one whole section of that one head with 2 tails is called the lipid. This is what we call the fluid mosaic model. And with this one, I'm not really concerned about it, but the function of vesicles and synthesis of proteins. Vesicles get the newly made proteins into the Golgi apparatus, and after the apparatus modifies it, the vesicles then transport them to other parts of the cell membrane where they are released.